All right, let's dive into a topic that's honestly mission critical for anyone who works with data: rigorous error control. We're going to talk about how we can be sure our data is actually telling us the truth. So let me ask you something. Straight up, the data sitting on your computer right now, can you really, fully trust it? In a world that runs on data, everything from complex microbiome studies to massive sweeping meta-analyses, the answer to that question changes everything.
Because here's the tough truth. If you don't have the right controls in place, your data can and will lie to you. It can send you down completely the wrong path, have you chasing ghosts, and really just undermine your entire understanding of what's going on.
So here's our game plan. We're going to start with the problem itself, then we'll define our solution, this idea of rigorous error control. After that, we'll break it down into 3 key pillars, see why this matters for your work specifically, and wrap it all up with the one big takeaway you need to remember.
Okay, first things first, let's talk about the problem with data. Because before you can really appreciate the solution, you have to have a healthy respect for just how misleading raw results can be.
You know, data is almost never perfectly clean, right? Especially in fields like biology. Sequencing data, it's just naturally noisy. And when you start pulling together these huge data sets, even the tiniest errors can pile up and become a massive problem. Then you've got things like lab contamination or batch effects. That's when experiments run on different days give you different results just because of tiny little changes in the lab, not because of any real biology. All of this can create false positives that can totally wreck your interpretation.
So if data is this messy, how in the world do we build reliable science on top of it? Well, that brings us right to the heart of what we're talking about today: defining rigorous error control. This is our solution to all that noise.
At its core, rigorous error control is a systematic process. And that word systematic is key. It's not some magic button you can just press at the end. It's a really disciplined way of thinking, where you're actively hunting down, modeling, and then cutting down on errors. And the goal? It's simple, but it's so important: making sure our results are accurate, reproducible, and trustworthy. That all sounds great in theory, right? But how does it actually work when the rubber meets the road? Well, it's all built on what you can think of as 3 main pillars of control. You know, a really good way to think about this is like quality control in a factory. They don't just check the final product as it rolls off the line. No way. They have checks at every single step of the process. Rigorous error control is exactly that before your data analysis pipeline. So these quality checks fall into 3 main buckets, or pillars. You've got measurement control, statistical control, and algorithmic control. Each one is designed to tackle a different way things can go wrong. Let's break them down. Pillar number 1 is measurement control. This is all about cleaning your data right at the source. We're talking about finding sequencing errors or mistakes from stuff like PCR amplification, where one little error can get copied millions of times. The whole goal here is to strip away all that technical junk so that what you're left with is the real biological signal you actually care about.
Okay, so once your data is cleaner, you have to be really smart about how you analyze it. That brings us to pillar 2. Statistical control. This is where you use powerful techniques like controlling the false discovery rate, or FDR. Think of it like a spam filter for your results. It helps make sure that what looks like an amazing discovery isn't just random chance, so you don't end up wasting your time chasing ghosts.
And finally, we have pillar 3, algorithmic control. Look, your analysis is only as good as the software you're using. This pillar is all about making sure your computational tools are doing what you think they're doing. Are your files getting corrupted when you move them? Is the algorithm itself introducing some kind of weird bias? This is about checking the integrity of the whole process.
Okay, so we've got these 3 pillars: cleaning the data, analyzing it smartly, and checking the tools. But let's bring this all back to the real world. Why does this actually matter for you and the work that you're doing?
Well, if you work in any of these areas, microbiome analyses, meta-analyses, multi-omics, this isn't just a nice to have. It's absolutely essential. The data in these fields is just unbelievably complex and messy, and errors can pile up so fast. Without this kind of rigorous control, your conclusions could be built on a foundation of sand.
And let's look at a really concrete example that the source material points out: a bioinformatics tool called Data2. It's used all the time in microbiome research, and its whole power comes from this really sophisticated error model that actually learns the sequencing error rates as it goes. It's a perfect real-world example of that first pillar, measurement control, in action.
Because at the end of the day, that's the whole point, isn't it? The ultimate goal of all this careful, meticulous work is to get to the truth.
Whether that's an accurate profile of a microbiome sample or some other scientific finding, it's about making sure your conclusions are based on reality, not just noise.
All right, we've covered a ton of ground, so let's bring it all home and focus on that final, most important takeaway.
If you only remember one thing from all this, make it this. Rigorous error control isn't a tool you run at the end. It's not a checklist. It's a mindset. It's a systematic process you apply at every single step, and it's designed to do one thing: make your results accurate, reproducible, and above all, trustworthy. So I'll just leave you with this final thought to take back to your own work. Now that you know about these 3 pillars and what to look for, I want you to take a good hard look at your own workflow and just ask yourself, what errors might be hiding in your analysis?